Welcome to motor development. Here we're going to study how we go from a sperm and an egg into a high-functioning physical being. And again, the emphasis here is on the physical. It's not on the cognitive, which they have whole other subjects for, particularly in the education department and the psychology department. So they're looking at the development of the self, and we care about that, but we emphasize here what's happening physically. I'll introduce you to this guy. This is Ashton, my grandson that lives in town. He just turned 6 months. Our daughter Tori and Jeff live in town, so we'll see a lot of him. And then my other two grandchildren live outside Chicago in Indiana, actually. And that is Colette, who is 2, and Beckett, who is 3 months. So we're going to get to actually see some of the developmental changes that happen in my favorite little human beings. What we would do in a lecture class now is have you describe to me the similarities between the very young and the very old. And after we talk about those, then I would ask the why. So what are some of the similarities? Well, here they both are kind of stooped, wider bases of support, usually have issues with balance, and they're going to widen the balance and then maybe hold on to something. So here the baby's holding on to a chair. He might need a walker, a cane, something like that. And all of this is to get to the point of physiologically what's happening to them, and that has to do with myelination. And so if they're demyelinating as they age and they're not myelinated yet as they're infants, all those, it's going to impact how our nerves take these impulses and tell our bodies how to move. As we look at the definition of motor development, you see that it's comprised of 3 parts here: growth, development, and performance. And it's across the lifespan. And what we're going to emphasize there is just that there's some complexity to it, and we want to look into some of that complexity. The first part here, growth, is just like it says, the word growth. One of these that's kind of interesting is obviously good nutrition and health and avoiding disease, all that kind of stuff is going to matter. But my neighbor gets really excited when she introduces me to her friends because I happen to be 6'8", and she thinks that's a big deal. She's like 5'2". And I just go to her, I go, Vera, I didn't do anything. There's nothing about it that is a characteristic that I can take pride in. It's genetic. I just grew this high because of my ancestors. Whereas other parts of it have to do with, did I practice the piano? Did I work hard at other things? Those are characteristics that I think there can be some pride in, etc., like that. So here, development is the change in the level of functioning. So I used to not be able to type, now I can type. And then physical anthropology, really what we're talking about is it's the study of human development, but it's the biological aspect of it, not so much the mental part of it, the psychomotor. We're going to touch on both. As we look at heredity and environmental factors, heredity is your DNA. It's the kind of body that you have. How many fast twitch muscle fibers do you have? How many slow twitch? How tall you are? Your general body type. Are you an ectomorph, mesomorph, endomorph? Those kinds of things. Your environment has to do with, was it a great environment for learning, health-wise, all of those kinds of things. And they're going to impact each other. And we're going to look at that closely in a minute. Heredity, the cells, the chromosomes that you have, your genetics, each chromosome of your 46 chromosomes contains approximately 1,000 genes. DNA is contained in the genes. And so that's the blueprint for what you're going to become. Maturation has to do with this internal timetable of these developmental events. One of the things that's so interesting that we need to remember is part of our DNA is how fast these things happen. And so we know that at 6 months, this percentage of kids can sit by themselves. And this percentage of kids can stand by, 1 or whatever. And so those characteristics are running off a blueprint that's a timetable. And that's really, really, really interesting as we look at infants. And it's going to tell us to some extent whether this child is developing normally or not. And we'll have more on this later, but this idea that there's a normal kid is not true. But I'll get into that. It's averages, okay? So most of us. So this leads us to this idea of nature versus nurture. And it doesn't have to be versus, it can be both. And so here, is it your DNA that made you the way you are, whether it's your temperament or whether it's your physical functioning, and this combination? So I picked out a few, I thought, somewhat profound quotes on this subject. While genes are pivotal in establishing some aspects of emotionality, experience plays a central role in turning genes on and off. DNA is not the heart's destiny. The genetic lottery may determine the cards in your deck, but experience deals the hand you can play. Scientists have proven, for example, that good mothering can override a disadvantageous temperament. So all of this is saying that you have the DNA, but whether or not that's expressed may be more up to you. Most of us experience gender conditioning so young, research shows it begins in infancy, that we misunderstand the relationship between nature and nurture, culture and biology, fitting in and being oneself. So what they're saying is we may think things are genetic, but we don't realize that we've already conditioned this individual a certain way. As I look at myself, I have 2 grandsons born during this pandemic. And so I sent them baby rattles, and the ones I chose were to amuse their parents. I really didn't think I was going to affect the child, but I gave them a baby rattle that's a little dumbbell, and I gave the other one the hammer of Thor. I thought the parents would find it funny. I think the kid will shake it and make noise. But these kinds of things, like the color you paint the baby's room, how you dress them, all of these kinds of things are impacting that child at some point, and we need to remember that. Another quote, on the whole, families with a history of schizophrenia seem more than 4 times as likely as the rest of the population to pass along the condition to future generations, even if, as ever, the illness rarely passes straight from parent to child. And I know some families with schizophrenia in their family, and so we know these genes are there, but why do they affect certain people and not others? Why does it skip the child and get the grandchild, even though those genes had to be in the parent to be able to pass them on? So there's a lot that we don't understand is basically what I'm trying to do. And then this one, the brain is heavily influenced by genes, but from birth through young adulthood, the part of the human brain that most defines us is less a product of genes with which you started life than of what life has thrown at you, because it is the last to mature. By definition, the frontal cortex is the brain region least constrained by genes and most sculpted by experience. This must be so. To be the supremely complex social species that we are, ironically, it seems that the genetic program for human brain development has evolved to, as much as possible, free the frontal cortex from genes. So this is huge, and you can't understand that yet because you haven't gotten the rest of these lectures yet. But what I'm going to tell you is that we start with a startup program. So you come with a set of plans that tell your body certain things, that you're going to have infant reflexes that are going to tell you how to eat, that are going to have a crawling reflex. You come built with that. But there's other parts that aren't that way. And what they're saying here is the frontal cortex is the most blank part of your brain. The most developed part is your midbrain. So it's like the top of your spinal column. It's just a big nub there. And that's going to give you life support. That's going to tell your heart to beat, your lungs to go, to eat, to have these things going. But you don't think when you're that little. You don't have thoughts. You don't say, oh, there's my dinner, I see a bottle, I'm going to go grab that and eat it. No, you have no idea of that. But you have a reflex that says if something touches my lip, I'm going to suck on it. And so these things are combined. And what they're saying here is because it is the last to mature, by definition, the frontal cortex is the brain's region least constrained by genes because the genes didn't set that up. It's going to be more impacted by the experiences it has. So this whole section was just to say, I want us to be amazed at what's going on, and I want us to start thinking about this idea that there's some things that are pre-programmed and there's some things that aren't. This filter, it's an old picture, and I looked to see if I could do a better one, but I just liked it. And so here, the heredity. So what's in your mind? What are some heredity factors? And so what I'm going to suggest here is, let's say a kid was born tall and athletic. Their parents were, or at least one was, or grandparents, somehow they got some genes that made them tall and athletic. So they have some fast twitch fibers, they have some coordination, that kind of a thing. Lifestyle, though, is you could have two of these kids that were both born tall and athletic, and one got a healthy diet and the other grew up with a sugar fast food diet. And so you know that even though the heredity, let's pretend here, was the same, the lifestyles aren't the same. So then as we look over here to the right in the picture, we see that these are filters that are going to allow us to have a certain amount of performance in our body. And so if we look at the top of also what's going in here, so let's say one of the kids went to basketball camp since they were 4. Every year they have these exposures to athletic type things, and they have the genetics for it, and they have a healthy diet for it. So they may become a fairly decent athlete. Let's say the other one still had the same genes, had a bad diet, and instead of their family being able to or being interested in sending them to volleyball camp, basketball camp, that kind of stuff, they were into computer games, they were into Xbox, they were into watching movies, they were into music, something different. And so their outcome's going to be different. So this just emphasizes it's not all about genetics. It's what we do with our genetics that's also important. And these slides we're not even going to look at, except I just need to make sure that I'm oversimplifying some of this. It's plenty complexity for what we need, but I want you to know that there's more and more that goes into this. This goes along with our definition of motor development. I'm going to ask you to reproduce this on a test. And the idea is that motor performance is impacted by a lot of things. So your motor performance is going to be different before you've grown, after you've grown, while you're growing, all of those kinds of things. Your genes, your maturity, all of these things, as well as your experiences, and then just the developmental stage you're in, all of these things are going to impact it. So it's just to try to emphasize the complexity. And now we get to a big chunk of this lecture and what we're after. There's 2 ways to study this, and one is stages and the other is phases. Stages, usually we go with chronological age, and the other we go by developmental milestones. And so both of these things are really interesting, and we kind of go back and forth, and it's not like they're very far apart. So I'm going to put this up on your exam, and you're going to fill in the blanks. But and this is very used, it's very popular to say, okay, what stage are you in? And so we say, look, we're going to take under 10s. And so we're going to use age as a way to group these children, and we're going to train them. So we don't want 14-year-olds in there. We don't want 8-year-olds. We want under 10s, and I guess an 8-year-old could be in there. But anyway, and we do this. We do it with Little League. There's an age cutoff, and then they go to Babe Ruth. And so age sometimes works well. And it's very easy. You go to the YMCA, how old's your kid? Boom, you're in a class. But sometimes it doesn't. So can you think this is where in class we would have a discussion and say, tell me something where we don't use age to categorize kids into training or learning or whatever. We certainly don't do it in college, right? So not all of you are the same age. Maybe you were closer in age when we started in kindergarten, first grade, you're within a year or two. But by now, there's probably a huge disparity in our class. And so I can think of Pop Warner football, if we were in a place where they had wrestling, These things, then they go by size and weight. So if you're in the 112-pound class, you're not going to hit somebody with, an opponent that's 175 pounds. So we categorize by size and weight when it matters. These stages, and here the only one that's hard, you could probably fill this in today just by thinking about it, is prenatal. I need you to realize that we're going to talk about things before they're born, that this is the developmental stage being prenatal. Then, and so filling this in, you're going to have the lines there, but remember to put prenatal before birth. Then infancy is up till they're 2, early childhood, later childhood, adolescence, adulthood, older adulthood. Okay, so right now you've got all those correct. Phases is probably going to be a little more for you, because before you're born, you're in a reflexive stage. That fetus is going through reflexive movements. It's kicking, it's yawning, it's doing all kinds of things inside mom. That's a reflective stage. After they're born, they're still in a reflexive stage. Sorry. And then we move somewhere around there into, we start blending these reflexive movements to rudimentary movements. And that's going to go until age 2. And so my grandson and some of the videos I'll have will have rudimentary movement. Reflexive and rudimentary is mostly what we're doing. Now, these aren't as easy for you, so you might have to practice a little bit. But you see, reflexive, we're going to 6 months. But rudimentary, you see, can start at birth and go to 2 years. So that's overlapping these lines a little bit. And that, again, it's not like a 3-stage rocket where the booster just falls off and you're into it. It's a blending. And so you blend from reflexive to rudimentary. All right. Fundamental movements from 2 to 6. And that's what's fun when we go to the early childhood center in my regular classes, and we work with 3 and 4-year-olds, and we teach them to throw, to kick, to catch, to dribble, all those kinds of things. And then sports skills, 6 to 12 years, and then growth and refinement, 12 to 18. Peak performance is where you are at, 18 to, let's say, 30. That's really interesting. When do people peak? And we'll have more on that in a second. Regression is definitely where I'm at, and so how are we losing our physical performance? And so these things are interesting. Here we would also ask you then in class, when do we peak? When do you peak physically? And it's debated because it depends on what we're talking about, right? It depends on in what way. But I'm going to tell you now, and it'll probably come up in another slide, but girls peak. Now, I'm not talking about you as an individual. I'm talking about females in the United States in general peak physically, and this is monitored on the, or evaluated on the 5 areas of physical fitness, endurance, strength, flexibility, agility, that kind of thing. And they peak around 12 or 13. And so if we were in class, we would talk about how many of you feel like you can run a mile as fast as you did when you were 12 or 13? How many can do as many pull-ups as you could do when you were 12 or 13? And so we see, now, this isn't necessarily a bad thing if we take into it consideration, meaning girls are supposed to put on weight after those ages, preparing them to be able to birth children. And hips are going to get wider, and so there's mechanical advantages, disadvantages going on there. But just weight sometimes has to do with that. Now, that doesn't mean you have to peak there. That means that all we're saying is that most females in the United States will never be as fit when we talk about physically functioning as they are when they're 12 and 13. They won't run as fast or as long or be as agile, any of those kinds of things. But we know college athletes, we know professional athletes can do that if they train to it. So part of it's physiology and part of it is our culture, and it's just interesting to think about when we peak. Males tend to peak, are you guessing? Somewhere around 18 to 22, something like that. And it depends on what we're looking at, but it is interesting to think that when you look at your insurance policies, an 18 to 22-year-old is the most, a male, is the most expensive insurance policy you can get when you're trying to get a driving or for auto insurance or something like that. Why? Because there's more accidents, because they take risk, they have poor judgment. And yet, these are the same people that we put into multi-million-dollar fighter planes because they have the reflexes, they have the physical strength, endurance, all those kinds of things to handle some of those pressures. And so maybe the brain isn't at its peak, but physically we probably are. Now, this used to be truer in that you wouldn't find very many Olympic athletes that are out of their 20s. And it depends on the sport. So I think Al Order still is the oldest in winning gold medals and stuff in the discus. Highly technical, high strength. Some of the weightlifting ones are you wouldn't find somebody in the 20s winning weightlifting. And so that leads us to, when do you peak? Well, we talked about your 20s, but you can peak with strength as old as your 40s. And so that's kind of an interesting concept, isn't it? To think about, you can be better than you've ever been in your life in your 40s. And then in my age group here in my 60s, I'm trying to not regress faster. So what about regression? Well, most people lose about 30% of your physical functioning between age 30 and 70. But you only have to lose about 15%. And that's what I'm trying to do, is try to minimize how much I'm losing. Okay? So as we look at the central nervous system, I'm assuming you all know this because you've all had anatomy physiology. You know what a neuron looks like. You know that there's an axon and multiple dendrites. You know the synapse is important to the connections between this, that the dendrites, the receptive part, there may be many of them. Do you know how many? We'll come back to that in a minute. Axon, though, there's only one axon. And then the myelin sheath. This is huge for us. It covers the neuron. And so we'll look at that in a minute too. So that's what's going to speed up our neurological processes. And again, like I mentioned early, the midbrain is the most developed at birth. It's like a big ball on the end of your spinal cord, and it has to do with life-sustaining activities, heartbeat, sucking reflexes, breathing, that kind of a thing. The cerebellum developed by 4, responsible for timing and coordination and balance, and the cerebral cortex by 2 years, some development, some voluntary movements, by 4, fully developed, responsible for thinking and reasoning. Okay, so we're not going to expect little kids to reason, but there's some going on. We'll get into it. So this is what I was trying to describe for you, and just how this is going to go. And I just want you to think about this developmentally, that this outer area is not, but the midbrain is developed. The cerebral cortex is going to be last, and this frontal lobe the last of the cerebral cortex area. Okay, this slide is going to show us about the sequence. And I don't know how I'm going to do quizzes and exams with you, because I don't know how I can get it so you can't cheat and all of those kinds of things. But what I do is I have you memorize this. And it is important, I think. And there's going to be some memorization on this first quiz, in fact, or exam. Most of it is memorization. But proliferation, migration, integration, differentiation, myelination, and necrosis, this is a developmental sequence that's going to tell us how these kids are going to develop and how we're going to move along from there. And then I need you to also be able to define each of them. So proliferation means generating new cells. Migration means these cells are going to move from one site in the brain over to another one that wants to be a motor cell, and another one's going to move over here that wants to be in charge of auditory or listening, and cells actually migrate to these different areas. Integration is how these neural groups start linking together, so that the eye area is also connected to the foot area, maybe through another area, so that they can communicate and work together. Differentiation is the progression of control, and all I care here is that you get gross movement to fine movement. And so that when we get through these stages, you're going to learn that the baby it's cephalocaudal, that it can first control its head, then its trunk, its arms, and out to its fingers, and then its legs. And so this gross to fine is what I'm after here in differentiation. Myelination, covering of the neural pathways, they become thicker, increases precision and speed. So if they're pre-myelinated, so we go to the early childhood center, they're not completely myelinated. So my catching group is going to throw a ball at this little kid that has their hands out in front of them like a cupped a receptacle. They're going to throw it. It's going to hit them in the chest, bounce off, then they're going to close their hands because they're not myelinated yet. So then instead of throwing, a hard ball or a tennis ball even, we might get a beach ball, something that is slower moving and bigger because they can corral it, they can group it with their arms, but they don't have the fine finger control to be able to catch it yet when they're 2 and 3 or 3 and 4. And then cell necrosis, we lose a tremendous amount without affecting performance. We lose 40 to 70% throughout our life from prenatal to death. There's no way to accurately measure all these kinds of things, and maybe you're thinking about football and you're thinking about other kind of head trauma injuries, but we just lose some of these cells. And so we want to keep them active and we want to keep them healthy as possible. There is a brain growth spurt, and that is from 6 months to 3 and 4 years age. So we're going to see huge changes in these kids, and that's why it's so fun to look at them. And then as you see here, if they don't get fed well, if they grow up in a home with smoking or other kinds of issues health-wise with an infection or whatever, this can hurt their development in certain ways, such as the number of neurons and the quality of myelin and such. Here, the brain changes in response to these experiences, and so we're going to get an increased weight and thickness of the cerebral cortex. Glial cells are the structural ones. There's more to them they're discovering, but they're the ones that are kind of the scaffolding, and they also help with the movement. Increase in size of the cell body of the neuron, and then increase in synaptic density and synaptic spines. So all these connections are going to happen. Okay, so how many other nerves can be connected to this one nerve? And so I'm telling you right here, it's more than one. So the idea here is one axon, but how many dendrites? And this is just one of those moments when we go, oh my goodness. And it's each neuron may be connected to up to 10,000 other neurons, passing signals to each other via as many as 1 trillion synaptic connections, equivalent by some estimates to a computer with a 1 trillion bit per second processor. So we're amazing. And all of this is taking place. So as I look at these infant grandchildren of mine, and I see this, I know that next week they're going to be able to do more, and the week after, and the week after. Indicators of brain development, we can't necessarily look in their head, but we can have them try things and see what's happening, and then we can guess what's going on. So as their reflexes get better, then we're assuming they're becoming myelinated, and that's a good thing. Nerve conduction, you see there for reaction time and movement time. Brain weight, the brain is only 25% of the final weight. By 6, nearly fully developed. Almost any alcohol contributes to low brain weight. And so all of these things are important. So we're going to test the child, and then we're going to make inferences about what's going on with their central nervous system. This is to emphasize the idea of myelination. So if the one on the left is myelinated and the one on the right is not, and so the impulse would go down the nerve, which seems like that would be easy and fast, but you know that as the myelin looks like sausages or something like that, that we end up with those nodes of Ranvier, and so then the impulse jumps, and so we can become, I believe it's 5 times, well, it says 2 to 3 times faster than a 5-year-old. And so that's really going to speed up things as we become myelinated. And then we're going to lose that as we age, and our reflexes are going to slow down. So here are 2 terms I need you to memorize for your exam, cephalocaudal. So cephala, it just means head, caudal means tail. And so this is where we control, and as the infant controls their body. And so they're going to control their head first and later the tail, but really we mean their lower part, their limbs, they're the legs, and it's going to go head to toe, okay? And then proximal distal is from your midline out to your fingertips. And this means that at first you can corral, you can make your arms go really wide, and your fingers may be doing something, but you can't pick up like a Cheerio yet. And so that's going to come later as they develop this other part. Mass to specific is again just gross to fine motor, bilateral to unilateral. So they'll use either hand until they decide. Now this is interesting to me because it says usually by age 6. Well, when we go to the early childhood center, we're going to test that, and so you should hand them whatever the implement is or something else. And the soccer people, the kicking people, they can make it so they can find out footedness. But the idea of handedness and footedness is, do they use their right hand or left hand all the time? And the people that have the hardest time, I think, has to do with the batting, and we call it striking. And so you'll set them next to the tee and you'll try to teach them how to swing, but you don't have a clue whether they're better at left or right, because there's not too many of them that had a lot of experience at that. And so one of them will swing and miss the whole tee and the ball, and then they'll come back the other way and nail it. And our people aren't ready for it because that seems so unusual to swing both ways like that. But to decide, the earlier you decide, the more athletic you're going to look, the more coordinated you're going to look. Because if you don't have to decide, oh, am I going to use my left hand or right hand? If you already know you're going to use your right hand, half of that decision making in your brain is already done, and you're good to go, and you're going to look so much better. Doesn't mean we can't develop our left side. We want to do that in almost everything, but our brain decides which one is the preference. So in our area and some sight people, there's a little argument there. We don't believe there's anything such as being ambidextrous. Not that you can't use both hands equally, it's where it's located in your brain. So if this motor center for this control is on your left side, then you're right-handed. And if it's on your right side, you're left-handed. And so that's all it's about, that there's going to be a center in your brain that's going to decide to take over. It's not whether or not you can dribble with either hand or something like that. Now, this is an older theory, this brain lateralization, and there's others, and the one I like more, it has to do with layers. Like there's an outer layer, inner layer, and this kind of a thing. But still, the idea is here. I'm just saying, don't take this as written in stone that can never change. But that we have areas of our brain that control different parts. And so here, this cross-section is the corpus callosum, and that's this bridge that connects the two. So hand and foot movements are controlled by opposite side hemispheres of the brain. In general, left governs language, logic, and sequential processing. Right is associated with music, emotion, spatial abilities, and intuition. The corpus callosum reaches adult proportions by age 10. This is important because as it gets thicker, you're able to share the information at an easier rate, and that's going to improve everything there. I've read more in the last year or two on this other idea of an outer layer and an inner layer that I like better than this, because we know this isn't true for everyone. The way they started this with electrodes in a net on your head, and they see what sparks when they get you thinking about music or whatever. And it doesn't mean there's not an area, but what I'm saying is it's not as adamantly done in stone as what some of the older books say. These two words I want you to be able to define, phylogenetic and ontogenetic. And the idea is you're going to say, I can't remember how I asked the question, but I ask you about learning or something like that. And then you'll say phylogenetic, but I need you to say things that acquire or that naturally acquired. So grasp, creep, crawl. So unless there's some kind of physical problem with the child, every group of people in the world walks. How does that happen? How come nobody developed to where they just crawl their whole lives or roll or do something else? Well, we think that goes off a blueprint of human DNA. We learn to stand up and we learn to walk. So those are phylogenetic skills or behaviors. Ontogenetic is not every human's going to learn to type. So these are skills that we learn probably because of our culture that they may have, we don't have, and it can be very technical things like we talk about, but maybe also it's carrying a big vat of water on your head, or something like that, that you and I might struggle with, but other people have learned this because of their culture. So I want you to say phylogenetic, naturally occurring, give me example, typing. Ontogenetic, occurs to all humankind, and walking, running, whatever. This is the origin of the words. There's different kinds of studies, and the longitudinal are best, so you follow individuals throughout their lifetime. But that's the most expensive, and it obviously takes a long time, and people want to get published before the kid turns 20. Here, cross-sectional ones have to do with, I'll take a group of infants, a group of teens, a group of 20, 30, whatever, and I'll look at all of them, and I'll see the differences, and I'll make my inferences from that. And then cross-cultural has to do with, I'm going to compare women in South America to women in Appalachia to women in the Sahara, and we're going to see the differences. So all of these are really interesting studies in motor development, and the one that we'll spend some time on is with infants, and how some of them are bound in certain cultures and in other ones they're not. Here, growth, it's a quantitative, how tall are you? So when I show you pictures of my grandchildren, it'll say, I think Ashton's was, he was in the 83rd percentile for height and 63rd for weight, or something like that. So it's just measurements, a mass and length, that kind of a thing. So how do we determine maturity? And so chronological age is easy, right? How many birthdays have you had? Biological age is a little bit harder, but the ones that we look at the most there are puberty. Have you gone through puberty yet? Skeletal age, as you see, I expanded this, can throw in some other things. And so here, I don't know if you how many x-rays you've had throughout your life, but I had a few when I was growing. And then some of these superstar athletes. When I was young in high school, I played against Bill Cartwright. He was from Sacramento area. And when I played him, I was a senior and he was a sophomore in high school. And so he was only 6'10" then as a sophomore. And at some point, I don't know when, if it was his junior year, senior year, some of the colleges, some of the pros, that kind of stuff, they wanted to see if he would continue to grow or if he was done. And so what do you usually do? They x-ray the long bones, and I think they did a wrist and a foot, and they see if there's still areas that haven't ossified yet. And they projected that he would be over 7 feet, and he ended up that way. And so we can tell these kinds of things somewhat by this, how the ossification is going. Again, this blueprint in your DNA that tells your body that this is how you mature. Now, we know there's a huge window there. We know that some girls enter puberty years before other girls, and the same with guys. And so we have to say there's a window there, but we want to start looking at these kinds of things. So we've already just compared this. There's over 800 growth center points in the body, 400 before birth, 400 after, mostly in the epiphyses of the bones. This is a fun little thing for me here. So this is our first grandchild. This is Colette. What's interesting to me, a lot of different things. First off, my son married very well. But Colette is in the 93rd percentile for height and 39th percentile for weight. Okay, that makes sense to me. But some of you are probably the opposite on some of those kinds of things. You can see Erica's dad and mom there. And so if you don't know me, I'm the one on the left here, and Scott and Kath are on the right. Scott is 6'6", and here he's had a stroke, he's had cancer, he's having a tough time. He's probably got less than a year to live in this picture. He was a Major League Baseball player for 19 years. He pitched mostly for the Cubs, but for the Angels he was in a World Series, all of these kinds of things. But here he's having some physical issues. In this stroke, you can see his ankle didn't work, so he has a brace on his leg and all these kinds of things. All right, so he's 6'6", I'm 6'8". Our wives are about the same height, somewhere, I don't know, 5'7"-ish. My son is about 6'3". Erica, I'm guessing, is 5'7", something like that. Now, what is the odds here of Colette she's probably not going to be tiny, okay? And she's probably not going to be real wide. But as we look at how the parents have passed, or I guess the grandparents in this case, have passed our DNA into our kids, and they get some of each. So my son Axel has some of my DNA, but he also has his mom's DNA, and the same with Erica. And then they pass their DNA into Colette. So how much of my DNA is in Colette? And the idea here is that some of you would say, an eighth or something. We have no idea. There is no way to know which genes got passed from me into my son and my son into her. We know the total that she ends up with, but we don't know which ones were selected for. Okay? But when you look at this whole thing of this, and so now I would like you to think about your families and how similar you are and everything. And so we do. We have families where everybody in my family, my dad's side had 9 brothers and sisters, so I have a million cousins. We have one male cousin that's under 6 foot. We don't know what happened to him, but it happens. But most of them are 6'3", 6'4". I'm the tallest at 6'8". Our girls are all 5'8", 5'10", 5'11", and then they married guys that are 6'3" and 6'4". So I have one cousin who is 5'11". She married a guy 6'6". They had 3 girls. So you know where this is going. One is 5'10", one is 6'1", and one is 6'3". And they didn't care for athletics, but they're great musically. But I got to tell you, the tall girl, she had such a hard time in junior high. She was so much bigger. Seriously, it was like Shaq going to junior high. She was bigger than her principal, all her teachers, everyone. And it was so hard on her emotionally that she ended up being homeschooled. And then she grew into this idea. But these can be huge factors in how your personality works out. I'm not going to try to get these running right now, but this is what I'm talking about, how we're going to look at my grandchildren. We're going to look at these developmental stages, and I have little YouTubes with this on these firsts. The other is I want us to realize that how long it takes from what I have is Ashton, I'm working on here still, is when he cannot sit up, and a week later he can sit up. And then I have that of my son, where he can't crawl, and then in a week he can crawl. And so these connections, it's moving, it's on a timetable, and these things are going to happen. That's just for cuteness. And the idea here is at 4 months she was at 93rd percentile. But do I think she's going to be over 6 foot? No, I don't think so. She's at 93% now, but she probably slow down. But she is a cutie.
This shows us where technology is currently with these micro cameras that can capture these kind of moments within our body. This is a picture of an egg being released. This is the moment of ovulation. We're going to start with the male side of this. A fertile male, when he ejaculates, can release between 2 and 5 milliliters of semen. Each milliliter normally has about 100 million sperm. If the concentration falls below 20 million, there's usually some trouble with fertility. While the male is producing millions of sperm, the female is producing normally 1 egg per month. This goes on till about 51. That's the average in the United States for menopause, but it could be in the 40s, it could be in the later 50s. So each time that she releases 1 egg, there's between 200 million and 500 million sperm trying to be the one that enters that egg and fertilizes it. Younger men have a better chance with healthier sperm, better swimmers, less damage to them, etc. So after 40, although men keep producing sperm, millions of sperm every day, they're healthiest before 40. But men never stop producing sperm. Here's a graphic to show all these sperm that made it to the egg, but once one of them buries their head into the egg, then a reaction is going to happen which releases these cortical granules that will prevent more sperm from entering the egg. This is a great picture. So first let's define what's going on. In artificial insemination, a sperm is placed in the uterus and conception happens otherwise normally. So other than the male doing it more naturally, a physician is putting it inside of the female. With in vitro fertilization, the eggs and the sperm are combined outside of the human body. This is actually a picture of my nephew's son or daughter. I don't know yet what it's going to be, but what they did was they waited for these cells to multiply, and then they chose some healthy ones. And I think in their situation, they only put in one, where in the old days, they'd put in many more than that, and you'd get triplets and such. But I think either one or two, but I think it was just one that they placed in there, and she's almost at the end of her pregnancy. This is to demonstrate there's similarities between other species. So if you choose E, I'm going to worry about you because I think that's from the movie Species. But the idea here is there's some similarities between different animals. So which one's human? Maybe you're looking at the head, maybe you're looking at the tail. I hope you're not looking at A, the little chipmunky thing there with the ears. But it ends up that C is the human. And as we look at another set, they're trying to show some more detail here, and that you see they all have tails, and they're different. Even humans have tails, and so sometimes they're born with the tail, sometimes it doesn't go past that. Now these tails aren't usually functioning tails; they're just like some flesh. I don't know if they have bones in them. Usually they don't. They just have some flesh, and they're removed when they're born. But these kinds of things and similarities. Now, why do we care about this? Because it may help with procedures such as using a heart valve from a pig or bovine, and so those kinds of things. This is another similarity. This is a human hand, obviously, and these are the bones inside a whale's pectoral fin, and it's interesting to see those similarities. Here you see a spinner dolphin at 10 months, great picture, and we think about how often they compare our brains with dolphin brains, etc. Here we see an elephant, 22 months, almost 2 years of pregnancy to bring this elephant into the world. And then here's a giraffe at 13 to 15 months. Maybe you've seen those cute National Geographic, etc. documentaries, and you actually see this giraffe and it falls like 6 feet to the ground, but pretty soon gets up on these wobbly long stilts and can move. And that's kind of the big deal here. What I'm trying to emphasize is that humans come out pretty much half cooked, meaning they're not ready to take care of themselves. They can't run away, they can't protect themselves. So our 9 months, our 40 weeks, is really only the beginning of the developmental stage. They can live outside, but not without a lot of help from mom and dad. This is to show some developmental stages along the way and how it turns from this thing that we can't really see much in to what ends up being a human. And I think one of the main things here is to see that the head is the most developed part during this time. And so if you have a brain stem that can go going, yeah, you need your heart and lungs, obviously, but do you really need your feet and hands and such, where they can develop later? And so you can see they're just kind of buds when they're born. Again, just developmental stages, real pictures. Here's a picture of a fetus, and it's 14 to 18 weeks, so it looks like it has a lot of detail, but there's still a lot to go. But again, the photography is great, and it kind of gives you an idea of where we're at developmentally. Now we come to, I hope I can simplify this so it's not hard, but it's definition of terms and the idea of pregnancy, fertilization, and conception. And so if we were in class, I would be asking you, what is fertilization, what's conception, what's pregnancy, and when do they start? And so what I'm going for is not a biblical situation. I'm going for a legal, scientific talk about this. And it's interesting to me because California changed this definition, and it's been a long time now that it did. But in the old days, people used to consider fertilization and conception synonymous. And you'll still see that in the writings. I was looking through articles preparing for this lecture this week, and you see them used interchangeably, and they're not, as far as legally and that kind of a thing. So fertilization, what is fertilization? The idea here is that fertilization is when the egg and the sperm meet. That's fertilization, okay? But implanting in the uterus, that's pregnancy. Pregnancy begins when this egg-sperm connection, and I'll give you some other names for it, is actually implanted. So pregnancy doesn't begin when the cells come together. It begins during implantation. And so what is conception? So some people will use it interchangeably with fertilization. I don't consider that accurate. To say it's the same as pregnancy, that's fine. And I'll tell you what all of that has to do here in a couple slides. So first off, fertilization occurs in the fallopian tubes. It's not in the ovaries. And so the sperm makes its way up there. The egg is released. The timing can be the egg is early, the sperm is late. So one of the things is sperm can live a few days in there. But the end here is once fertilization takes place, this newly fertilized egg is called a zygote. From here, the zygote will move down the fallopian tube into the uterus. The zygote then burrows into the uterus lining. This is called implantation. When it implants, it's called a blastocyst. That's the beginning of pregnancy. Fertilization doesn't always occur even if the egg is released. A fraternal twin occurs when two eggs are released during this ovulation and both eggs are fertilized. So they're not going to look like each other because it's two different genetic patterns. Different eggs, different sperm. Identical twins are when a single embryo splits after fertilization, resulting in identical twins, because both cells come from the exact same egg cell and sperm. Identical twins will have the same DNA, the same sex, and nearly identical appearance. You see that on crime shows occasionally where one of them committed a crime and left DNA, but they can't tell which of the brothers did it. Pregnancy begins only once the embryo is successfully implanted into the uterus wall. Emergency contraception isn't the same as abortion. So this is where I'm getting into these definitions, and this is going to be a really big deal to some people, but it just for this. So a long time ago, when fertilization and conception were the same, when the egg and sperm met, that was fine. But then when they came up with more and different times of contraception, then contraception means it prevents conception. So if the egg and sperm already met, you can have those kinds of contraceptions that don't allow that to happen. And so there are those. But now they move that technical term to during implantation is conception so that you can have the day after pill and these kinds of things that will stop this this group of cells from implanting into the uterus. So now that's contraception. So conception is implantation. You stop that. You're still legal. You can buy this at drugstores, all of those kinds of things. Different churches have different views on this, and I'll get to that a little bit. An ectopic pregnancy is when the fertilized egg implants outside the uterus. Usually this is in the fallopian tubes, and it can be very dangerous for mom. Pregnancy tests detect the hormone level, and it's human chorionic gonadotropin. And as those levels double every 2 to 3 days in the early stages of pregnancy. And so that's what they're measuring when you take a pregnancy test. Here, we're learning that pregnancy doesn't start when the egg and sperm come together at fertilization. It actually starts the first week is counted from the first day of your last period. So what that means is you have 2 weeks when nothing's happening inside you, but that's still part of your pregnancy count. And that's where they get to 40 weeks for the whole gestational term. And this is just to say that there's a difference between an embryo and a fetus, and that ends up being at 8 weeks. The embryo ends and the fetus begins. Getting back to the California type stuff, in California, it's legal to get an abortion for any reason until approximately 6 months after pregnancy begins. So we're going to look at the first trimester, second trimester, and so before the third trimester there. After that, if pregnancy puts the mother's health or life at risk, an abortion can still be performed. Tons of court cases in states at the federal level, all of these kinds of things have been hashed out. Back in the day, Bill Clinton's era, that kind of a thing, it was legal to have partial birth abortions. So as long as the whole baby didn't come out, the doctor could still go in and make it so the baby couldn't live. Then there were things like, could it live outside the womb? But eventually it's come to where most of the states that allow this, it's at the third trimester. This is within 24 hours after fertilization, and so the egg begins dividing rapidly. This is day 4, so this hasn't implanted yet, it's still tumbling down the fallopian tube. So what are some of these implications? The question of life when begins, and however this separate but closed question of when the woman is considered pregnant, medical community has been clear. Pregnancy is established when a fertilized egg has been implanted in the wall of a uterus. It's critical to distinguishing between a contraception that prevents pregnancy and abortion that terminates it. On this point, federal policy has long been consistent and in accord with scientists. Drugs and devices that act before implantation prevent rather than terminate pregnancy. And then again, we see here an interesting one. The establishment of pregnancy takes several days and is not complete until a fertilized egg is implanted in the lining of a woman's uterus. Pregnancy is considered to be established only when the process of implantation is complete. Everything is individual, though. Women just have fewer chances to conceive at the beginning and end of a cycle. But look at fertilization occurs within 12 to 24 hours after ovulation. Only during this short period, ovum stay alive. As you see, when absolutely healthy couples don't always conceive during the first few months of pregnancy planning. The most widespread time for implantation is the 9th day after ovulation. Nearly 40% of women have conceived on the 9th day after the moment of their ovulation. How long does implantation take? 6 to 7 day trip. It's dividing. It's undergoing different stages. The zygote begins a morula. The morula becomes a blastocyst. Another 2 to 3 days, the future embryo is moving freely in the uterine cavity, finishing preparation for implantation. Women sometimes know when this happens because there can be some spotting, or they can start feeling sick, again, as these hormones change. Progesterone levels are going to change, and then some women get early pregnancy cramping. So again, just to summarize this, the fertilization is the joining of the sperm and the egg, and this does not necessarily happen at the moment of intercourse. This can be within days. So the sperm can stay alive. Usually they say 5 to 7 days, something like that, 6, 7 days, but as few as 2 to 3 days, these things can happen. So it can take time to get up there. When people, maybe you've seen some of the things funny in movies or whatever, when a couple's trying to have a baby, sometimes then they'll have intercourse, and then she'll kind of like do headstands or something like that to help gravity and everything and help the sperm move to where they want it to go. During the trip, it'll be the zygote, the morula, and the blastocyst. Twinning. So there's different kinds of twinning we know, but when the division of the developing zygote into 2 embryos occurs, 99% of the time it's within 8 days of fertilization. I don't know when this is going to come up in the other lecture, but we'll just talk about it now. This is interesting to me because I really like talking about how different churches, denominations, religious groups look at these issues. And the one that is the most analytical, most academic are the Jews. And the Jews say they don't care what you do in the first 7 days because they don't believe whether you call it a soul or a spirit, inside this egg and sperm here, they don't believe there's a spirit in there or a soul because it could still twin. And they don't think there's two souls in there that are going to split. They don't think you can split a soul. So they say anything before this must not have a soul. Then after this, either they've split or they haven't, and they have a soul enter them. So that's how they come up with it on when to them the life begins, and to them it's the spiritual life. Ovulation is in the middle of the menstrual cycle, 28-day cycle, subtract 12 to 17 days from the end. It could be a week either way. Women, some are very regular and some are not. It could be 2 ovulations and have multiple eggs. Fertilization needs to take place within 12 to 24 hours of the egg being released. 40% will implant on the 9th day. The length of the pregnancy is 280 days or 40 weeks. And the first day of the last menstrual cycle we talked about last period, and then this estimated date of confinement. However, only 4% of women actually give birth on that day. 2 of those weeks are before fertilization of the 40 weeks. Okay, just some quick pictures, and then you can see on the side some of the little things that are going on. This is to create a sense of awe of what's going on. And 7 weeks, a human kind of appears, maybe a Martian type looking human. But you can see how it's the size of a kidney bean, and you can see all the vessels and bones starting in there. 12. It's about 3 inches long, weighs nearly an ounce. Her tiny, unique fingerprints are now in place. This is about 11 pennies. The baby is now 5 inches long, weighs 5 ounces. His skeleton is starting to harden from rubbery cartilage to bone. 6 to 7 months, the baby weighs about 1.5 pounds. His wrinkled skin is starting to smooth out as he puts on baby fat under the skin. At 8 months, it weighs about 4 3/4 pounds. His layers of fat are filling out, making him rounder, and his lungs are well developed. Complete at 9 months and ready to be born. In 40 weeks, what was once a cell is changed into a human being. Again, this is to emphasize the idea that developmentally, they look at it in trimester. I'll show this. But week 1 and 2, you're getting ready. Week 3 is fertilization. Remember, week 1 and 2 is the mom is getting the right environment for the egg. Then fertilization, 3, implantation, and that's when pregnancy begins. And then the embryonic period begins in week 5. Neural tube closes in week 6. So these are the kinds of things those apps will tell you every week, and maybe help you be really good with your nutrition, etc. Baby toes, all of this kind of thing. Fingernails. So then we get to week 40, and it might be that it goes past that. It might be early. So what can go wrong? There's 3 areas that I want you to be aware of, and I'll have some questions on. Congenital, genetic, and chromosomal. And all I need you to know is how they're different. And so congenital is one of the ones that I really think you're going to have more trouble with, because it means there's no genetic problems. Everything that mom and dad gave them, all the DNA, all of these kinds of things are perfectly fine. And they paired up and did everything. But during those developmental stages that we just saw, maybe it's week 7, maybe it's week 8, something didn't go the way they normally do. And so here, some of the ones that are common, if you could say common in an abnormality, is a hair lip, a cleft palate, heart defect, spina bifida, absent limbs, or extra digits. I knew a girl that had 6 fingers, but the 6th one wasn't useful, and they ended up just cutting it off. So as we look at this, now here, when we look at the cause of spina bifida, so the genes are there, they're fine, but maybe what it takes to support those genes isn't there. And so here, research studies indicate that insufficient intake of folic acid, a common B vitamin, in the mother's diet is the key factor in causing spina bifida and other neural tube defects. So it doesn't close all the way. And just giving her a little bit of vitamin and this wouldn't happen. Now, that's in general. They also think, though, that there might be some, if you see here, multiple genetic, nutritional, and environmental factors. So the environmental might be German measles, it might be something like that. But the genetic part too is maybe you need more folic acid than the next person. And so it's when all of these things come together. But we know it's important for a mom to have enough B vitamins, and that's why they have pregnancy vitamins for them to take, so that they'll be making sure that this isn't the cause of a defect in their child. Severe congenital abnormalities such as major heart and central nervous system malformations might result in a miscarriage or death of the baby before week 24. And these defects can almost always be picked up with ultrasound screening. Here's a little one that's just kind of a funny one where she has webbed toes, and she used to be self-conscious about it, but then she decided to embrace it and just put a little tattoo there. This is trying to teach you more. We're going to come back to this in another part, but the length of your index finger and ring finger are determined by the amount of testosterone exposure and when. So it's when you get exposed to it and how much. And so you can see the ring finger in A is longer, and in B the index finger is, and in C they're the same. Does this matter? No. But at the same time, the same dosage of testosterone is also in the brain, and that can affect different parts of your brain because you're feeding it more of this powerful hormone. Well, I'm changing up now to heart defects, and I just love that video, so I wanted to use it. So again, we're on congenital abnormalities, and here's one with the bicuspid aortic valve. And this is close to home for me because my daughter had this. And so something happened before birth as the blood vessels and other parts were forming, and it didn't exactly get together. But you see the one above that? It might be present at birth, but it wasn't there when they were born, although it may not affect them until years later. So here in the center is the bicuspid valve. And I love this. It's just one of my favorite things because right here, the blood in the heart doesn't nourish the heart. And so here, when it squishes and all the blood comes shooting out of this, it's at the highest force anywhere in the body is right here. And it's so powerful that the pressure here in this hole would rupture this vessel if it went directly in there. And so the valve opens and closes that little hole as it opens with the rush of this blood. And then when it relaxes, the backfill of the blood can go fill the heart. And so I just love that. But anyway, you see it's a tricuspid valve if it's working correctly. Our little girl who ended up, she was an All-American in track and field here at Point Loma. She's in the Hall of Fame. And she had a congenital heart defect. And this is after her running career. Did the running affect it? We don't know. Did it wear it out? We don't know. But we knew there was a heart murmur type thing in junior high. Every year she went in and they listened to it and did it. Then after college, she had some symptoms of being tired and when she worked out. And so you can imagine what it's like. She goes to the doctor and says, well, I'm supposed to have my heart checked every year and I haven't had it checked. She went to law school and did some other things and she got off the rhythm, and then she changed insurance, medical issues. So she wasn't with us anymore. She was on her own. And so she's trying to get them to say, no, I need to get this checked every year. And they go, well, why? What are your symptoms? He says, well, I'm tired after I run a couple miles. And they go, well, everybody's tired after you run a couple miles. And so anyway, they finally did. And she looked fine. She killed the stress test. They cranked it up as high as she could. She did fine. But then as it was coming down, the elevation of the test, then they were doing an echo and they were looking at it and they go, wait, stop, get off immediately, all this kind of stuff. And what was happening was her valves weren't attached like they should. So it squishes out, but it was like a flapper. And so it let it come back the other way too. And so it was very inefficient. And so sometimes a tricuspid valve has a bicuspid and they get together, and then sometimes a vibration makes it kind of calcify and get hard. Hers was a mono. It was just flapping back and forth. So they got her in as quickly as they could to do surgery. This is the great nurse in the ICU and monitoring all of this while she's there. This is kind of what they found, and so here I have had as many physiology courses as you had probably, and here I have never heard of a left atrial appendage, but she has one, and you're born with it, whatever. But they don't like it because blood can come in here and kind of pool, and then if blood pools, it can coagulate and throw off clots. So while they were there, that's not why they went in. They decided to close that off, and this is what they did. So here she has a bovine valve. This doesn't mean they take the valve out of a cow and put it in there. It means they use uh, the pericardium of the cow to make their own uh, tricuspid valves. And then they're on the shelf and they have different sizes. And you can have a 22 mil or a 24 mil. And the bigger ones, the better. And so when they went in, they felt her artery was pretty small, her aorta. And so they slid it, put Teflon in as a patch, and made it bigger so it would accept a bigger valve. And so then they placed this valve in there, and that's what you see here in this X-ray is the wire of the valve frame. This bobby pin looking thing cuts off that atrial appendage, and so it just closed it off with that. What they did for her surgery, as you've seen those people that get their chest split open, they have that big scar all the way up down there, and they crank open their chest. And the day before her surgery, when they were in there, the surgeon says, "Hey, you want to try something new?" And he'd never done it before. So she's in her 20s, and they cut her across below her breasts, lifted all of that tissue up, and then sliced open her chest. Then they put her back together with all these wires. Then they put back her breasts and sewed it up. So if she's wearing a sports bra, that big band at the bottom is where her scar is. And so she doesn't have the cosmetic issues that might be, a little uncomfortable for some people, especially younger people. And the doctor was thrilled that it worked so well. And this is her chasing this manta ray in Hawaii, and this is 3 months after her open heart surgery. So it went pretty well. Okay, we're switching to genetic abnormalities, and this is just what you're thinking. There's a problem with the gene. I don't think these others are as hard to understand as the other ones are. So here, cystic fibrosis is the most famous, and it's because it's one gene. And if you have this one gene, they can easily see that you're going to have it or that you could carry it, and so it's a big deal. So these are genetic abnormalities that we know there's sex-linked conditions such as muscular dystrophy and hemophilia. This graphic is to help us understand how sex-linked characteristics can be passed on or not passed on. So here, mom has the bad abnormal gene. It's the yellow one, the yellow X. And here, she gave the good one to this unaffected female. She gave the good one to this unaffected male. She gave the bad one to the carrier female. And she gave the bad one to this affected male. So the top two siblings have no problem because they got the X from dad, and so that's fine. And there was no, it's not a carrier or anything else. This is a healthy female. She's not going to pass on this trait. Here's a healthy male. He doesn't have the defective gene anywhere inside him. He has a healthy one from mom, a healthy one from dad. This is a female character, a carrier. She's not going to be affected by it because she has a healthy X from dad, but she has the defective one from mom. So if she gives this defective one to her son or daughter, well, to the daughter, she'd be a carrier, and to the son, he'd be affected. And that's what's happened down here in this last child. That got the affected one from mom, and dad didn't give the X to cover it, and his Y won't cover it because it's on the X chromosome. So this kid will have hemophilia or some other kind of genetically sex-linked problem. Okay, chromosomal abnormalities. Probably the first thing you think of is Down syndrome. We know it's a big deal. They can just look at what happened there and see that. The other one that's big is dwarfism, and I think with the cable shows and such, that's become much more popular. But there's causes more than 200 conditions, and it causes proportionate dwarfism and hormonal metabolic issues. And you know that most of these shows, they're going in for surgery or some other kinds of things, and their lifespan is usually shorter than other people. Epigenetics is a word you need to know, and what it can mean is that you have the genes, but whether those genes are expressed or not is a big deal. Nutrition can affect this, but so can other type of things like stress and your environment, whether it's in utero or after you're born. Why your DNA is not your destiny is pretty interesting. So this is just what I'm talking about. A plant-based diet might be the answer, and it's going to help you. We'll talk more about that. Here we can see the size of the head is the first thing that's developed, and then it's only a quarter of your body when you're an adult. If that's probably less than that. And so just to see that where you're a newborn and, oh, at the newborn, it's a quarter. And as an adult, it's a little pea on top of that big body. So here's a newborn with a lot of cartilaginous areas. Here they're measuring height and weight, but this is an interesting thing that they project how to measure it for old people that are all bent up. And what they do is they take the length of the tibia, and then they go from there. They take the knee height and their age, and they can approximate what it would be. We know there's some differences, and I know there's some ethnic differences in different peoples on how long limbs are, but it's an interesting way to, if you've been around particularly old women who have osteoporosis and they just shrink, to see where they would have been or were. This just gets into those kinds of things, and all this is for right now is to say we're going to see that it's a scheduled growth, and that we look at these weeks and we see what's happening in those weeks. It's just to show the schedule. You don't have to memorize these things or anything. And that then there's special times like these. We all know about puberty when people just shoot up in size, but there's a mid-growth spurt between 5.5 and 7 years for most people. My kids, when they started eating a ton, we knew they were in a growth spurt. Here's other factors: heredity, hormones, nutrition, disease, and climate. Now, climate is the weird one, right? Everything else you would guess would affect all of this kind of stuff. But it ends up, puberty in hot climate areas is much earlier and faster than in cold ones. So girls' puberty age could reach 8 or 9 years in hot climates. Kind of interesting. Here we start exploring the idea of puberty and what affects the difference in some people's going into puberty earlier or later. And we've already kind of mentioned climate might do that, but there's more that we need to at least give some kind of thought to. Does it really matter is a big deal, and that's why we're going to go through this fairly quickly, but there's some things that are pretty interesting. And so here the dip isn't much, from 2 months to a year, depending on the study. No one yet knows why, although there's a number of intriguing theories, nor even whether it's a problem. So here, girls typically develop breast pubic hair between 10 and 11. African American girls tend to do it a year earlier. But is it because they're African American, or is it because of overweight and obesity? So we know fat is related to this, and fat is also a source of estrogen, and so all of these things might happen. Now, there are people that think the longer you're exposed to estrogen, that increases your risk of breast cancer, so it may matter in some ways. Here we find family structure in Arizona. They looked at young girls who spent more time living in a home without their birth fathers were more likely to go through puberty early. Animal studies suggest that exposure to pheromones of biological fathers appear to slow down puberty in females, but other factors could be at work. And so here another one was divorced families, and the father was high-risk behavior, meaning violence, depression, imprisonment, and then left the home. The younger the sisters in those families tended to go through puberty nearly a year earlier than the older ones, indicating that emotional stress at a certain period in early life may affect a girl's normal development. So all of this is to say, just like we started earlier, there's a lot of factors that influence our. They looked at cow's milk to see if those, and nobody thinks that that is a factor of cow's milk. Uh, there are many more health consequences to being overweight than having your period at 12 instead of 12 and a half. And for some tweens, having a physical appearance that is more mature than their emotional age can also be a problem when it comes to navigating the world of boys and dating. So, should they go by age? Should they go by emotional? This slide introduces the idea of melatonin and how does melatonin affect your internal clock and calendar? Well, active kids produce more melatonin. And so if they're heavier kids, they go through puberty sooner. Is this a problem? They don't know. This one has to do with pollutants like PCBs in their food, and some of these, a lot of pollutants are stored in fat, and so that's a double whammy there if you're heavy and you have pollutants, and these might be tricking kids' bodies into going through puberty prematurely. Again, the cold temperature, does that delay, and does hot temperature speed up? The reason I go through those quickly is because it's interesting, but those are tangents for where we really want to go. I think you need to know the generals here and know that your hormones, whether it's testosterone, estrogen, progesterone, all of these things are going to cause you to grow, and we want those to be in normal ranges at normal times. The adrenal cortex, the androgens above the kidney, also important, as well as pituitary gland, human growth hormone at the base of the brain. So all of these things are going to kick in and tell us when to grow. Males have approximately 10 times the level of androgens than females, and therefore males grow larger. Estrogen inhibits growth. Females have this, but also have testosterone. And so after menopause, women's hormones are going to change, and their ratio, testosterone, is going to be higher compared to estrogen. So some people need uh, uh, hormone replacement therapies to balance that out. But women might have a tendency to grow a little mustache and to put fat on their belly where they used to put it on their hips because they're a higher percentage or ratio of testosterone. Now we're turning into the past of puberty and looking at males and females. And so males tend to grow over a longer period of time. And that means that females are easier to predict their growth because it's in a shorter window. The head is a quarter of the body size at birth, an eighth of the body size at adulthood. Your center of gravity lowers with age. Well, they're assuming that you're going to put on some fat around the middle. Males are leaner at all ages, have a greater body density, so more muscle. Those that mature earlier tend to be taller and heavier for that age. Females are taller and heavier than males until puberty. This is fun to look at the early childhood center. The biggest kid in the 3 and 4-year-olds has pretty much always been a girl, but most of them are fairly interchangeable there. Females have earlier and smaller growth velocities, so they don't shoot up 6 inches like a guy can in a year or something like that in general, but I'm sure it happens. At puberty, males increase in muscle cell production, and at puberty, males start having more lean body mass. We already mentioned that females are more easily predicted just because it's smaller, not as radical jumps. The secular trend I want us to understand, so here in class we would talk about, are you taller than your parents? Do you think your kids will be taller than you? And so a secular trend means generation to generation, are we getting taller? Well, that was true for a long time, but then it slowed. And so the deal is, if you come from a place where you got bad nutrition, or some of the Southeast Asian countries where they didn't get much protein and lived on rice, and then their kids have better nutrition, they're going to be probably bigger than their parents. But after a couple of generations or so with good nutrition, it shouldn't matter. So we're not necessarily getting bigger. But one of the things I usually like to ask in class, if anybody's been to England, where they have these suit of armor, and the guys in there were tiny little guys wearing these suits of armor, and so we are bigger. I believe the tallest country is still the Netherlands, and then the Scandinavian countries are up there. I know there's a tribe of Watusis in Africa that have 7-footers. But as far as a whole country, I think the Netherlands is still the tallest. I don't have any idea who the smallest people are. We're also maturing earlier. Every 10 years has decreased by about 4 months of menarche, the first period. Indicators of brain development. So we can't look in there, so we're going to see how nerve conduction is and our reaction time and our movement time and the size of our head, especially little ones as they're growing, and they're going to actually measure their head and hope they don't have too big, like hydrocephalics, or too little. And then this last section here that we're pulling up is on nerves. And the idea here is just that they've been doing some research on these different kinds. I'm sure these look familiar to you. You got the dendrite, the axon, but there's more kinds of than just. And so here we have the unipolar neurons is what we typically see, but there's bipolar neurons with dendrites at both ends, and then there's multi. And that's fine, but this is what I really want to show you. That here they're talking about dogs and they're smelling, and the ones that have the same type of nerves, they don't smell as much different things, but if they have a whole bunch of different varieties of nerves, then they can smell a lot more scents. And so it's good to have a variety of these. Here they're able to map the nervous system of a rat, and it's just showing where technology is and how we're progressing. And again, we want to talk about how the midbrain was developed at birth, and then by 4, we're starting to get real thinking into the kids. But over and over again, we'll be looking at these kids' videos. They're not thinking they're not making sophisticated plans on that they need to. Our grandson, he's on a clock, and when he gets hungry, he just starts screaming. When he sees a bottle, he gets excited. So at 6 months, he does know that that bottle is going to feed him. But he'll sit there and he'll open his mouth, but he won't reach for it with his hands. He doesn't have a plan on how he's going to satisfy all these needs. You're probably familiar with homunculus man, and the idea here is these proportions are to where the closeness of the nerves, the sensitivity of the nerves. It's interesting. I would have thought genitalia would have been a little bit bigger there, but the idea of this. Now, why do we care? I care more about these associations on that, where they might be in your foot, your iris, your ear, those kinds of things. But here, what they're trying to show us is if we were to put this in a sensory map of the brain on where these things would be. Okay, this is the end of this lecture. This has been the prenatal development. Next up is going to be infant reflexes. I know this is long, it's almost an hour and a half. I will watch the time. I'll make sure that it's not going to be any longer than it would have been in lecture, but it might not be split in exactly hour or 50-minute periods.